Hi viewers, welcome to SEKHAR'S Bio Factory for NEET Principles of Inheritance and Variations with Codominance concept with 5 examples. If you like this video, subscribe my channel and click on bell icon to get notifications. Like, follow and share my videos to your students and friends to get more information in biology related to NEET and other national state level competitive exams. In this video, I am going to explain the codominance concept with 5 examples in plants, animals and human being. Codominance is a type of dominance where the offsprings are newly produced progeny. It shows similarity to both the parents and it is due to allelic interaction present on homologous chromosomes. Codominance is observed when two different alleles of a specific single gene trait are both expressed simultaneously. Such type of codominance it follows non-Mendelian inheritance pattern. The offsprings will be a combination of both the parents. Codominance is a form of inheritance where in the alleles of a gene pair in heterozygous condition are fully expressed. Codominant expression occur when two different alleles are inherited at the same genetic location like capital A allele, capital B allele. In this allelic pair, one of the chromosome it is called father chromosome, that is paternal chromosome. Another chromosome it is maternal chromosome or mother chromosome. Both are present in the form of pair. In such condition, the allelic pair came from both father and mother. It result in the product of both of the alleles. It will be expressed. Two alleles for a trait are equally expressed with neither being recessive or dominant. It creates codominance. The alleles present on homologous chromosomes they fail to mask the another allele of the same gene. Codominance occur when the phenotype of both parents are simultaneously expressed in same offspring organism. Some important examples for codominance. The first example is flower color in Camellia plant, seed coat nature in lentil plant, hair color in cattle, AB blood group in human, MN blood group along with sickle cell anemia. These are the examples for codominance concept. Let us discuss the first example, that is petal color in Camellia plant. Flower color in Camellia plant called Camellia japonica is one of the example for codominance. In Camellia, the red color petals denoted with capital R in flower and white color petals are denoted with double capital W. It expresses white color petals. The heterozygous condition capital W express both red and white color petals. It result in a flower with alternating patches of petals in a flower. That means in heterozygous condition, the both parental characters are equally expressed so that the flower contain white and red color petals alternatively in the form of patches. It shows codominance. The heterozygous progeny only it shows codominance. The inheritance pattern of petal color in Camellia japonica. In this inheritance pattern, by conducting a cross in between red flower and white flower plant denoted with capital R, capital W, capital W. At the time of cross, both parents can produce only one type of gametes. The first parent can produce the gametes with capital R. Second parent can produce gametes with capital W. These gametes are fused to form F1 generation. In F1 generation, the plant contains flower with the red and white color patches alternatively having a genotype capital R, capital W. Again, by conducting a cross in between two F1 hybrids or in between two plants with red-white petals, it can produce two types of gametes. In these, 50% of gametes having capital R allele, 50% of gametes having capital W alleles. In such cross, by conducting a cross in between two F1 hybrids, F2 generation is formed with different combinations. The first combination, the plant having only red color flowers because of capital R allele. The second plant having red and white color petals alternatively because of capital R W alleles. The third combination also having red and white color petals alternatively because of capital R W alleles. The fourth combination, the plant having white color petals because of capital W allele. In this way, in F2 generation, totally 4 offsprings are produced. In this offsprings, one is with red color flowers, two are with white red color petals, and one plant with white color petals. When we calculate its phenotypic ratio and genotypic ratio, the phenotypic ratio is 1:2:1 because red is 1, red white are 2, white is 1. In similar way, the genotypic ratio also it becomes into 1:2:1 because capital R is 1, capital W is 2, capital W is equal to 1. In this way, both phenotypic ratio and genotypic ratio it becomes into 1:2:1. It is the first example for codominance. The second example, seed coat nature in lentil plant. Seed coat nature in lens culinaris is another example for codominance. It is simply called lentil plant. In lentil plant, the seed coat having patches. So these are the patches present on seed coat. It is expressed due to capital CS alleles. In some of the seeds, the seed coat contain dots. This dot character is expressed because of capital CD allele. These are the two traits observed in seed coat nature in lentil plant. At the time of heterozygous condition, if the gene having capital CS, capital CD alleles, it shows both patches and dots. So both patches and dots are observed on seed coat because of codominance. When we observe the inheritance pattern of seed coat nature in lens culinaris plant, by conducting a cross in between two parental plants, in this one of the parental plant having seeds with patches on seed coat, another parental plant having seeds with dots on seed coat denoted with capital CS, capital CD alleles. During gamete formation, both parents can produce only one type of gametes because of homozygous condition. The first parent can produce gametes with a capital CS allele. Second parent can produce gametes with only capital CD alleles. These gametes are fused to form F1 generation. In this F1 generation, the F1 hybrid, it contains both patches and dots on seed coat because of heterozygous condition denoted with capital CS, capital CD alleles. This F1 hybrid can produce two types of gametes. In this, 50% of gametes are with the capital CS alleles, 50% of gametes are with the capital CD allele, and by conducting a cross in between two F1 hybrid, we get F2 generation. In this F2 generation, the first combination, the plant having only patches on seed coat because of capital CS alleles. The second plant having both patches and dots because of capital CS and capital CD alleles. The third offspring also having both patches and dots on seed coat because of capital CS, capital CD alleles. But the last combination, the fourth plant having only dots on seed coat because of capital CD allele. In this way, we get F2 progeny. In this F2 progeny, when we calculate phenotypic ratio, the seeds with patches on seed coat is equal to 1. The seed coat with the patches and dots plants are with 2. Only dots with 1. That's why the phenotypic ratio it becomes into 1:2:1. In similar way, the genotypic ratio of codominance is also equal to 1:2:1 because capital CS allele is equal to 1, capital CS CD is equal to 2, capital CD is equal to 1. In this way, in lentil plant related to its seed coat nature, both phenotypic ratio and genotypic ratio it becomes into 1:2:1. Coming to third example, it is related to animals, hair color nature in cattles. In codominance, a heterozygous individual expresses both simultaneously without any blending. An example of codominance is the roan cow, which has both red hair and white hair on its body surface. A cow having white hair on their body because of capital RW allele. Now it is crossed with a bull having red color hair on its body surface because of capital R smaller alleles, and by conducting a cross in between these two, in F1 generation, due to heterozygous condition, the offspring has both white and red hair called roans with a genotype capital RW, capital R, small r alleles. The inheritance pattern of skin hair color in cattles. By conducting a cross in between a cow with white color hair denoted with RW, capital RW, and bull having red color hair because of capital R small r alleles. During gamete formation, these individuals can produce only one type of gametes. The first parent can produce only one type of gametes with capital R W. Second parent can produce only one type of gametes with the capital R small r alleles. When both gametes are fused to form F1 generation, in this F1 generation, because of heterozygous condition, both alleles express its character so that the cow or offspring having both white and red color hair on their body surface, such individuals are called as roans. This F1 hybrid roan can produce two types of gametes. In this, 50% of gametes are with the capital RW, 50% of gametes are with the capital R. Now, these two types of gametes are fused randomly to form F2 generation. In F2 generation, the first combination is with white color hair because of homozygous capital RW, capital RW. The second combination it becomes into roan because of heterozygous condition capital RW, capital R, small r. The third combination also it becomes into roan because of heterozygous condition capital RW, capital R, small r alleles. But the last combination it becomes into red, having red hair on body surface because of homozygous condition capital R alleles. When we calculate the phenotypic ratio in this, white, roan, red is equal to 1 is to 2 is to 1. In similar way, the genotypic ratio capital RW is to capital RW, capital R is to capital R is equal to 1 is to 2 is to 1. The next fourth example, it is the AB blood group in human. The codominance means that neither alleles can mask the expression of another allele. In this, an example in human would be ABO blood group where allele A and allele B both are expressed in AB blood group. So, if an individual inherits allele A from their mother and allele B from their father, they have a blood group AB. That is example for codominance. Out of 4 blood groups, only AB blood group is example for codominance because remaining A, B, O blood groups it shows Mendelian inheritance pattern, but AB blood group only it shows codominance. The inheritance pattern of blood group in human. Blood group A and blood group B. By conducting a cross in between two persons, especially father with blood group A, mother with blood group B, in these individuals, the blood group nature, it is heterozygous in nature. The father with the blood group A having alleles capital IA, small I. Mother with the blood group B having capital IB, small I alleles. In a cross in between father and mother, it results in the formation of two types of gametes. The father with the blood group A can produce two types of gametes. In this, 50% of gametes are with the capital IA allele, 50% of gametes are with the small I alleles. Mother with the blood group B, due to heterozygous condition, here also two types of gametes are produced with the equal ratio, that is 50% of gametes are with the capital IB allele, 50% of gametes are with the small I alleles. These gametes are fused randomly with Punnett square method. At this time, after fertilization, we can observe different combination. The first combination is capital IA, capital IB alleles. Because of capital IA, capital IB allele, both alleles they expressed in progeny. Due to this codominance, AB blood group will be expressed. So only AB blood group is an example for codominance. The next second combination is capital IB, small I allele. Due to dominance, only IB allele it will be expressed and progeny gets B blood group. The third combination is capital IA, small I. Because of dominance, capital IA only expressed and the progeny gets blood group A. The last combination is small I. Because of a homozygous recessive condition, the progeny will get O blood group. So in this way, we can observe 4 types of blood groups when we conduct a cross in between heterozygous parent with the A blood group and B blood group. In such case, in heterozygous condition only 4 types of blood groups are possible in progeny. The next example, Mn blood group in human. Mn blood group is a special type of blood group. The Mn blood group system is under the control of an autosomal locus found on chromosome 4 with two alleles designated with a capital LM and a capital LN. These alleles controls Mn blood group nature. Landsteiner and Levine discovered Mn blood group system. The blood group type is due to a glycoprotein present on the surface of red blood cells which behaves as a native antigen but antibodies are completely absent. Antigen M present in M blood group, antigen N present in N blood group. Both antigens are present in Mn blood group due to codominance. The inheritance pattern of Mn blood group. A cross in between two persons with M group and N blood group. The M group is expressed due to capital LM alleles and N group is expressed due to capital LN alleles. In this cross, both parents can produce only one type of gametes. In this, first parent can produce one type of gametes with a capital LM. Second parent can produce only one type of gametes with capital LN. Both gametes are fused to form F1 generation. In this F1 generation, because of codominance, both alleles will be expressed so that the blood group becomes into Mn group. And this F1 hybrid can produce two types of gametes. In this 50% of gametes are with the capital LM and remaining 50% of gametes are with the capital LN. These gametes are fused randomly to form F2 generation. In this F2 generation, the first combination is M group because of homozygous capital LM. The second individual shows Mn group because of heterozygous condition with capital LN. The third combination it becomes into Mn group because of heterozygous condition capital LN. The last individual shows N group because of homozygous condition capital LN. In this way, the three types of blood groups are expressed in F2 progeny. When we calculate the phenotypic ratio, it is equal to 1:2:1 because 1 M group, 2 Mn group, 1 N group. The genotypic ratio also it becomes into 1:2:1 because the capital LM is to 1 is equal to 1, capital LN is equal to 2, capital LN is equal to 1. In this way, both phenotypic ratio and genotypic ratio it becomes into 1:2:1. These are the 5 examples. Along with these 5 examples, we have to discuss one more important inheritance pattern, but it is related to inherited diseases like sickle cell anemia. The next example, it is sickle cell hemoglobin. It is not a common method in all individuals. That's why we discussed at beginning with the 5 examples, but it is a special method, a special pattern of inheritance present in only sickle cell anemia patients. Codominance. It refers to the expression of polymorphic alleles resulting in a new phenotype. The altered form of hemoglobin. This altered form or mutated hemoglobin, it causes sickle cell anemia, is inherited as a codominance trait. A person, if it having the two alleles for hemoglobin, it is HbA, normal hemoglobin molecule is produced. But some of the persons, if it have HbS alleles, they have sickle cell hemoglobin and it causes sickle cell anemia. In the sickle cell anemia, biconcave RBC or red blood cell, it becomes into elongated sickle shape. The heterozygous individuals with the HbS alleles, these individuals express both normal and sickle hemoglobin, so they have a mixture of normal and sickle red blood cell, shows codominance. In the inheritance pattern of sickle cell hemoglobin, by conducting a cross in between two individuals, both individuals are carrier for sickle cell anemia because HbA allelic combination, it can produce normal hemoglobin. HbS alleles with heterozygous condition can produce or it becomes into carriers. The homozygous HbS, it becomes into sickle cell anemia. So these are the three types of allelic combination for normal, carrier and sickle cell anemia person. But in this cross, we are going to take the two individuals with the sickle cell carriers having HbS alleles. By conducting a cross in between two carriers, these are heterozygous individuals. Because of heterozygous condition, the individuals can produce two types of gametes. In these, 50% of gametes are with the HbA alleles and 50% of gametes are with the HbS alleles. These alleles are fused randomly to form different combinations. At this time, to observe its combination, we have to take one Punnett square. During fertilization, when we observe the first combination, it is a normal hemoglobin because of homozygous condition. In this combination, HbA alleles are fused to form HbA allelic combination. Because of homozygous condition, this person gets normal hemoglobin molecule. The second combination, it is example for codominance. In this, due to fusion of gamete with HbA allele, the combination it becomes into HbS. It denotes heterozygous condition. In this heterozygous condition, due to having of HbS alleles, both alleles are equally expressed, so that in that individual, because of heterozygous condition, it becomes into sickle cell carrier. In similar way, the third combination also, because of heterozygous condition, that is HbA and HbS allelic combination, in such condition, the heterozygous individuals are produced. These heterozygous individuals acts as a carriers. But finally, due to fusion of gametes with HbS allele, now this allelic combination, it becomes into homozygous. It causes sickle cell anemia. Because of sickle cell anemia, the individual may be die. It is the inheritance pattern of sickle cell hemoglobin. In this condition also, by conducting a cross in between two carrier persons, we get 1:2:1 as a genotypic and phenotypic ratio. In this, the phenotypic ratio is calculated as out of 4 individuals, 1 individual is normal, 2 individuals are with the carriers, 1 individual with affected. The genotypic ratio also, it becomes into HBA-HBA1, HBA-HBS2, HBS-HBS1, so that the genotypic ratio, it becomes into 1:2:1. So same sickle cell anemia concept we will discuss in upcoming videos with the complete information. It is the inheritance pattern of sickle cell hemoglobin molecule. Out of 5 or 6 examples, the first 5 examples are normally present in plants, animals and human beings. But sickle cell anemia, it is the special condition present only in sickle cell anemia patients. All these are the examples for codominance concept. Thank you for watching. If you like this video, subscribe my channel, like my videos, share my videos to your students and friends.